My name is Dr. Abdul Wahab, Associate Professor, Department of Pharmacy, Quart, University of Science and Technology. Our today topic is Niosome as a drug delivery system. As you can see over here in this picture, it is the structure of Niosome. So in Niosome, mostly in the preparation of Niosome, non-ionic surfactant, cholesterol, and charge inducers are used. So you can see over here, it is the hydrophilic part and it is the hydrophobic part of non-ionic surfactant. Uh, it is the aqueous compartment of niosome. So before going to discuss in detail, these are the different contents. We will try to discuss all about these contents like definition, advantages, disadvantages, structure and drug encapsulation, types and size of niosomes, composition of niosome, factor affecting niosomes, methods of preparation of niosome, difference between niosomes and liposomes, applications, marketed products. So these are the contents for our today discussion. So first of all, we would like to discuss about the definition of niosome. That word are niosomes. So as you can see over here, they are lamellar microscopic structures which are produced by a non-ionic surfactant, cholesterol, admixture, and a charged inducers or molecules with a subsequent hydrating in water media. So you should keep these three component uh, if you are going to prepare a newsome like non-ionic surfactant, cholesterol, and charge inducers. Why we use non-ionic surfactant and why we use cholesterol and charge inducers in the preparation of niosomes. So as you can see in the structures, it is a structure, it is a picture of niosome, and you can see over here, uh, it is the tail of non-ionic surfactant and it is the head of non-ionic surfactant. So the head part of niosome, as you can see here, it is a hydrophilic part. Which part? Hydrophilic part of niosome. So as we discuss that in the composition or in the preparation of niosome, non-ionic surfactants are used. So it is the hydrophilic part of the non-ionic surfactant and it is the hydrophobic, we can say lipophilic part of the niosome. It is the tail and it is the head of non-ionic surfactant. So it is the aqueous compartment, and the heads of the non-ionic surfactants uh, also form the center, uh, the core of the niosome, as you can see over here. If we are going to load the hydrophilic drugs or lipophilic drugs, so we can easily incorporate the hydrophilic drug in this area, and similarly in this area the hydrophilic head of the non-ionic surfactant, and similarly if we want to load the hydrophobic or lipophilic drugs, so we can easily embed or incorporate the hydrophilic drug in this region of the niosomes. So it is the, as you can see over here, it is the hydrophilic head and it is the hydrophobic part, or we can say the hydrophobic part is the tail of the niosomes. So inshallah we are going to discuss the difference between the liposome and niosome. So one thing you should keep in your mind. So in a non-ionic surfactant, the non-ionic surfactant contains single tail. But if we are going to prepare the liposomes, in liposome, mostly we use the phospholipids. So the tail of the phospholipid contains two heads. So we can say there are two tails in phospholipids, while there is single tail in non-ionic surfactants. So niosome have a hydrophobic and hydrophilic moiety infrastructures, as you can see, like this, which allows drug molecules with a large range of solubilities to be accommodated. So large range, we can say that if we are going to load the different types of drug like lipophilic drugs, hydrophilic drugs, so we can easily load such type of drugs on niosome. So niosome were first reported in the 1970s as a feature of the cosmetic industry by Willenborg and Henjani Villa. The first niosomes were formulated using cholesterol and single chain surfactants such as alkyl oxyethylene. So you can say, so the first, so in the first niosomes, so cholesterol and single chain non-ionic surfactant like you can see alkyl oxyethylene was used. Niosomes are non-ionic surfactant based unilamellar or multilamellar vesicles in which an aqueous phase of solute is entirely enclosed by a membrane resulted from the organization of surfactant macromolecules as a bilayer. Understand? Niosomes are a novel drug delivery system in which the medication is encapsulated in a vesicle composed of a bilayer of non-ionic surfactant active agents. The particle size range from 10 nanometer to 3000 nanometer. The particle size range from 10 nanometer to 3000 nanometer. And you can say that niosomes are microscopic, bilayer, uh, microscopic vesicles uh, which are having the size from 10 nanometer to 3000 nanometers. But one thing you should keep in your mind that the difference between liposome and niosome is that in case of niosome, we mostly use the non-ionic surfactant, cholesterol, and charge inducers. While in case of liposomes, instead of non-ionic surfactant, phospholipids are used in the preparation of liposomes. But liposomes are also the microscopic vesicle, bilaminar vesicles, similarly like niosomes. But the difference is that in case of liposome, phospholipids are used. And the other difference is that the tails of phospholipids are having, or we can say phospholipid consist of two tails, while the non-ionic surfactants consist of single tails. Advantages of niosome. Niosomes are used for a variety of drugs, includes hydrophilic, lipophilic, as well as amphiphilic moieties. So for such type of drugs, we can use the niosomes as a drug delivery systems. And niosomes act as a depot to release the drug slowly and offer a controlled release. If we are going to prepare the slow or sustained or controlled release formulations, So we can also use the niosomes for such type of drug delivery systems, drug delivery formulations. And niosomes are osmotically active and stable. Niosomes increase the stability of the entrapped drugs. If we want to uh, increase the stability of the drug, so we can easily entrap or encapsulate such type of drugs which are having low stability, or those drugs which are not stable. So we can enhance the stability of such type of drugs by means of in if we are to prepare such type of and niosome formulations. Handling and storage of surfactants don't require any special conditions, as we discuss that in the preparation of niosome, non-ionic surfactants are used. So handling and storage of non-ionic surfactant, or we can say the surfactants, don't require any special conditions. Enhance the skin penetration of drugs. If we want to enhance the penetration of drugs through skin, so we can formulate or incorporate such type of drugs in niosomes. Disadvantages of niosomes. Niosomes are physical instability. Niosomes are physical instable, and there are chances of aggregation and fusion. Similarly, leaking of entrapped drugs, hydrolysis of entrapped drugs, which limits the shelf life of dispersion. So there are chances of hydrolysis if we are going to load the drugs on niosomes. So there will be chances of hydrolysis. So these are the disadvantages of niosomes. Structure of niosomes and drug encapsulation is, you can see over here, it is the structure uh, of a niosome, the hydrophilic part and hydrophobic part of niosome. Niosomes are formed from the self-assembly of non-ionic surfactants in aqueous media, resulting in closed bilayer structure. As you can see, it is a closed bilayer structure of niosomes, unilaminar or multilaminar, capable of encapsulating both hydrophilic and lipophilic substances. So, niosomes are having the capability to encapsulate hydrophilic and lipophilic substances. The assembly into closed bilayer is rarely spontaneous and, usually involves some input of energy, such as physical agitation or heat. Physical agitation or heat. Niosomes are microscopic lamellar structure which are formed on the admixture of non-ionic surfactants of the alkyl or dialkyl polyglycerol, ether class, and cholesterol with subsequent hydrations in aqueous media. Subsequent hydration in aqueous media. So we can say that for the formulations of niosomes, aqueous media is mandatory. So as we discussed in our previous slides, that the composition of niosome is non-ionic surfactant, cholesterol, and charge inducers. Niosomes may be unilaminar or multilaminar depending on the method used to prepare them. So the different methods which are used for the preparation of niosomes. So we will discuss the factor affecting on the formulation or on the niosomes. The hydrophilic ends are exposed on the outsides and inside of the vesicle, while the hydrophobic chains face each other within the bilayer. Hence, the vesicle holds hydrophilic drugs within the space enclosed in the vesicle, while hydrophobic drugs are embedded within the bilayer itself. So types and their typical sizes. Sizes of what? Sizes of niosomes. So these are the different types and their sizes of niosomes. You can see small unilamellar vesicle size is from 10 to 100 nanometer. As you can see over here, it is the unilamellar niosome. So large unilamellar vesicle, as the name indicates, large unilamellar vesicle, the size is from 100 through 300, 3000 nanometer. It is the small one and it is the large one. It is the large unilaminar vesicle. Multilaminar vesicle size is more than 3000 nanometer. These are the different lamella, as you can see over here. First lamella, second one, and third one. So due to different lamella, it is called multilamellar vesicles. Composition, as we have already discussed about the composition of the niosome, but for your kind information, these are the different constituents which are used in the preparation of niosome. The major components used for the preparation of niosome are cholesterol, non-ionic surfactant, charge groups. Cholesterol is used to provide rigidity and proper shape. If anybody asks you that why we use cholesterol in the preparation of niosome, so your answer should be that due to use of cholesterol, we can enhance the rigidity of liposomes and proper shape, conformation to the niosome preparation. In the bilayer structure of niosome, cholesterol form hydrogen bonds with the hydrophilic head of the surfactant. The role of surfactant play a major role in the formation of niosome. So the different non-ionic surfactants are used in the preparation of niosome. Niosome non-ionic surfactants are amphiphilic molecules that comprise two different regions. One of them is hydrophilic, water soluble, and the other one is hydrophobic, organic soluble. Non-ionic surfactants are a class of surfactant which have no charge groups in their hydrophilic heads. They are more stable and biocompatible, cheaper, and less toxic compared to their anionic, amphoteric, or cationic counterparts. Charge molecules, these are also used in the preparation of niosome. Charge molecules increase the stability of the vesicles by the addition of charge groups to the bilayer of the vesicles. So how the stability is increased if we are using the charge molecules? So there will be no coalescence and aggregations if we are going to incorporate in the preparations of niosome charge molecules. They increase surface charge density and thereby preventing vesicle aggregation. Prevent vesicle aggregations. Why we use the charge molecules? To enhance the stability. How? To prevent the vesicle aggregations. And this prevention is possible if we enhance or increase the surface charge density by means of charge molecules. So diacetyl phosphate DCP and phosphatidic acid, being an ionic in nature, are negatively charged that induce negative charge. Sterile amine STR, also known as octadecyl amine, and sterile pyridinium chloride, being cationic in nature, are positively charged that induce positive charge. Charge inducers are added to stabilize niosomes. To stabilize niosomes. So these are the different factors affecting physico-chemical properties of niosome, like choice of surfactants, nature of encapsulated drugs, either it is hydrophilic drug or hydrophobic drugs, and similarly concentration of cholesterol and methods of preparations. So these are the different factors which are affecting the physico-chemical properties of niosome. So choice of surfactant. A surfactant used for preparation of niosome must have a hydrophilic head and a hydrophobic tail. The hydrophobic tail may consist of one or two alkyl or perfluoroalkyl groups, or in some cases a single steroidal group. The ether type surfactants with single chain alkyl tail is more toxic than the corresponding dialkyl ether chains. The ester type surfactants are chemically less stable than ether type surfactants, and the former is less toxic than the latter due to ester linked surfactant degraded by esterase to triglycerides and fatty acid in vivo. The surfactants with alkyl chain length from 12 carbon 12 to carbon 18 are suitable for preparation of niosome. Similarly, nature of encapsulated drugs. The physiochemical properties of encapsulated drugs influence charge and rigidity of the niosome bilayer. The drug interacts with the surfactant head groups and develops the charge that creates mutual repulsion between surfactant bilayers and hence increase vesicle size. The aggregation of vesicle is prevented due to the charge development of bilayer. So these are the different factors which are affecting the physico-chemical factors of niosomes. As you can see in these pictures, the different factors: inclusion of charged molecules, nature of surfactant, structure of surfactant, temperature of hydration, and nature of encapsulated drugs. So these are the different factors which are affecting the physico-chemical properties of niosomes and niosomes formations. Concentration of cholesterol. The concentration of cholesterol is also having effect on the physico-chemical property of the niosome. The concentration of cholesterol depends on HLB, hydrophilic balance wheels of surfactants. The surfactant lipid ratio is in the range of 10 to 30 millimole and a 1 to 2.5% weight by volume. It may influence the physical properties and structure of niosomes. Appropriate quantity of cholesterol increases the hydrodynamic diameter and entrapment efficiency. A high concentration of cholesterol decreases the release rate of the drugs due to increase in the rigidity of the bilayer. So the surfactant and lipid ratio or so cholesterol ratio should be like this: 1 to 2.5% cholesterol must be used. So methods of preparation are also having effect on the physico-chemical properties of the niosome. Different methods are used to prepare the niosome suspension, or we can say the niosomes. Reverse phase evaporation method gives small size vesicles. Yeah, we will try to discuss the different methods which are used for the preparations of niosomes. So reverse phase evaporation is also the method for the preparation of the niosome. So reverse phase, if we are going to prepare the niosomes by means of reverse phase evaporation methods, so in this case, so the niosome size will be smaller. Hand shaking method give larger vesicle than the ether injection method. Ether injection, hand shaking, these are the different methods which are used for the preparation of niosomes. Niosomes prepared by transmembrane pH gradient method gives niosomes with greater entrapment efficiency and better retention of entrapped drugs. If we want to prepare those niosomes in which which are in which the entrapment efficiency should be increased. So which method should be used? Membrane transmembrane pH gradient method. It is the best method if we want to enhance or to increase the entrapment efficiency of niosome. Methods of preparations. So these are the different methods which are used for the preparation of niosome. Injection method, thin film hydration method, sonication, reverse phase evaporation method, the bubble method, microfluidization method, multiple membrane extrusion method, transmembrane pH gradient, drug uptake or drug pH gradient method, formation of niosome from pronosomes. So these are the different methods which are used for the preparation of niosome. The first one, injection method. So there are two types of injection methods. One is ether injection method and the other one is ethanol injection method. So as you can see, our slow injection of an ether solution of niusomal ingredients into an aqueous medium at a high temperature. So, as we have discussed in our previous slides, that for the formulation of niusomes, non-ionic surfactant, cholesterols, and charge inducers are compulsory, are mandatory. So, after the mixture of such type of the constituents or the ingredients, so aqueous medium, the addition of aqueous media is also necessary if we are going to prepare the niusomes. So a mixture of surfactant and cholesterol is dissolved in ether. For example, you can see 20 ml and injected into aqueous phase 4 ml. Aqueous phase is mandatory. It is compulsory for the preparation of niosome using a 14 gauge needle syringe at 60 degrees centigrade. So niusomes in the form of large unilaminar vesicles are formed. If you want to prepare unilaminar vesicles, but such that this type of unilaminar vesicle must be large, so then we should use ether injection method. And it is another injection method, that is ethanol injection method. Ethanol injection method offers advantage that it avoids both sonication and high pressure. In this technique, surfactant dissolved in ethanol and forcefully injected in aqueous media using syringe. The whole system stir using magnetic stirrer in order to evaporate ethanol, which results in formation of niusomal vesicles. So these are the pictures or diagram of the ether injection method. You can see over here, and it is the ethanol injection method. It is the ether injection method and it is ethanol injection method. It is the picture of uh, ether injection and ethanol injection methods which are used for the preparation of niusomes. Thin film hydration method. It is the thin film hydration method for the preparation of niosome. Thin film hydration is one of the most widely used method for the preparation of liposomes and niosome. This method could be also be used in the formation of niusomes. It is a simple method which involves dissolving the membrane forming materials in an organic solvent in a flask. As you can see, these are the different flasks over here. In a flask, as shown in the figure, after removing the organic solvent by vacuum evaporation, a layer of dried thin film forms inside the flask. Like you can see, a layer of thin film is formed inside of the flask. The drug is dissolved in aqueous solution such as water or buffer, like you can see over here, buffer, and then added to hydrate the dry film. Which film? This one. It is incubated about the transition temperature of the surfactant in a water bath to form niosomes. Like you can see over here. These are the different niosomes which are formed by thin film hydration methods. Niosomes for prepared by thin film hydration methods are multilaminar vesicle. So one thing should keep in your mind, if you want to prepare multilaminar vesicles, yani we can, if we want to prepare multilaminar niosomes, so this method must be used. Sometimes this technique is used together with sonication to acquire niosomes with narrow size distributions. This method is widely used to formulate niosomes loaded with drugs such as insulin, doxorubicin, and other extracts. Sonication method. Two techniques of sonication are employed: bath sonication and probe sonication, as shown in figure over here. It is a bath sonication and it is a probe sonication. Probe sonication technique, drug dissolution in phosphate buffer solution is added into cholesterol non-ionic surfactant mixture. The whole system is allowed to sonicate for 3 minutes at 60 Celsius degree centigrade through probe sonicator, which results in nano-size vesicle. Probe sonication is one of the techniques to reduce the particle size of niosome. Bath sonication, it is done after film hydration technique for achieving prolonged release and longer duration of action. These are the diagrams, you can see over here the different pictures of probe sonicator and bath sonicator. Reverse phase evaporation technique, which is used for the formulations of niosome and cholesterol. Liposome. In this method, cholesterol and surfactants are dissolved in a mixture of ether and chloroform. Mixture of ether and chloroform. Which component should be dissolved? Cholesterol and non-ionic surfactants. An aqueous phase containing drug is added to these and the resulting two phases are sonicated at 4 to 5 degrees centigrade or Celsius. The clear gel formed is further sonicated, as you can see, aqueous drug solution and surfactant and cholesterol in organic solvent. Which type of organic solvent? Ether and chloroform. So after the addition of aqueous drug solution, two phase system, two immiscible phases, you can see this one, the aqueous phase, and the upper one is surfactant and cholesterol containing the organic solvents like ether and chloroform. The organic phase is removed at 40 degrees centigrade, or we can say Celsius, under low pressure, as you can see over here. The resulting viscous niosome suspension is diluted. It is the viscous niosomes suspension is diluted with phosphate buffer saline and heated in water bath at 60 degrees centigrade for 10 minutes to yield niosomes, as you can see over here. Solvent evaporation under reduced pressure. Which solvents? Like ether and chloroform. These are evaporated and niosomes are prepared. The bubble method. It is the bubble method which is used for the preparation of niosome. It is novel technique for the one-step preparation of liposomes and niosomes without the use of organic solvents. This method can also be used for the formation of liposomes. The bubbling unit consists of round bottom flask with three necks. 1, 2, 3. These are the three necks of round bottom flask. Position in water bath. It is a water bath to control temperature. Why we use the water bath? Water bath is used for the controlling of temperature. Water cooled reflux and thermometer are positioned in the first and second neck. It is first neck and that one second neck and it is the third neck of the uh, round bottom flask. And nitrogen supply through the third neck. Nitrogen supply. Cholesterol and surfactants are dispersed together in the buffer. Which type of buffer? The buffer which is having the pH 7.4 at 70 degree centigrade or Celsius. The dispersion mix for 15 seconds. This dispersion with high shear homogenizers and immediately afterwards bubble at 70 degree centigrade or Celsius using nitrogen gas. It is the method which is used for the preparation of niosome and liposome. Microfluidization. It is the microfluidization techniques as you can see in this picture. So, which is used for the preparations of niosome, and this method can also be used for the preparation of liposome. This is a recent technique to prepare small multilaminar vesicles. Multilaminar vesicles. A microfluidizer is used to pump the fluid at a very high pressure through a screen. It is then forced along defined microchannels which direct two streams of fluids to collide together at right angles, thereby effecting a very efficient transfer of energy. The lipid and surfactant can be introduced into the fluidizer. The fluid collected can be recycled until spherical vesicles are formed. So we can say that for the preparation of niosome and liposome, we can also use the microfluidization technique. And there is one another technique, that is multiple membrane extrusion method. The multiple membrane extrusion method is also used for the preparation of what? For the preparation of niosome and liposomes. As you can see in this diagram, it is the different units of the multiple membrane extrusion method. In this method, mixture of cholesterol, non-ionic surfactant and diacetyl phosphate is solubilized in organic phase and further evaporated to produce transparent film. The film is dried in desiccator followed by hydration using aqueous phase containing drug. The whole system is then extruded via a series of polycarbonated membrane as shown in this figure. Polycarbonated membrane. The resulting suspension contains vesicle of uniform size. So, shortly we can say that multiple membrane extrusion method can also be used for the preparation of niosome and liposomes. Other methods which are used for the preparation of niosome and liposome: transmembrane pH gradient drug uptake method and formation of niosome from pro-niosome. These are also the methods which are used for the preparation of niosomes. Applications of niosomes: antineoplastic age treatment. Niosomes are used in antineoplastic treatment, leishmaniasis treatment, studying immune response, transdermal drug delivery system, niosome as a drug carriers, ophthalmic drug delivery, drug targeting, and these are the different methods which these are the different applications of niosomes. Marketed products, these are the different marketed products. Those products which are approved by the FDA and available in the market, like you can see over here, Lancome. Lancome has come out with a variety of anti-aging products which are based on niosome formulation. Uh, Lumigon ophthalmic solution by allergen containing bimetoprost for open angle glaucoma. Transport ophthalmic solution by Merck containing dorzolamide hydrochloride for open angle glaucoma. Estee Lauder Beyond Paradise Aftershave solution by Estee Lauder. Neoactive potent antioxidant and skin moisturizer by Naturals Life Technologies, and other products are also available on the market. So you can also search the different marketed products of Niosomes. Evaluation of and characterization of Niosomes. The different if we want to prepare the different we want to prepare Niosome or liposomes. So after the formulations, different techniques are used for the characterizations. Like if for the size, we can use DLS, SAM, FM, STM, CL. These are the different techniques which are used for the size determination. Similarly, zeta potentials, DLS and electrophoretic mobility, encapsulation efficiency. How can we determine that how much drug has been encapsulated, or how much drug is loaded on the neopsome or liposomes? These are the different techniques which are like HPLC, UV visible, and fluorescence techniques. These are the different techniques which are used to prepare to determine the amount of the loaded drugs in on the liposomes. Stability, how can we study the stability of the liposome, DLS. So it is also the diameter laser scatter which is used for the preparation of the neosome. Difference between neosomes and liposomes. So these are the differences between the neosomes and liposomes. You can say less liposomes are less expensive and liposomes are more expensive. Liposomes, these are the differences between niosome and liposomes. Chemical stability, we can say nisomes are more stable as compared to liposome. Chemically, liposomes are unstable. Nisomes are prepared from uncharged single chain surfactant, and liposomes are prepared from double chain phospholipids. So double chain means that liposomes, the phospholipids are having two tails, while the phospholipid, while the surfactants, non-ionic surfactants, are having single tail. They do not require special storage and handling. New somes don't require special storage and handling. While in case of libosome, they require special storage, handling, and purity of nature for phospholipids is variable. Non-ionic drugs carriers are safer. The ionic drug carriers are relatively toxic and unsuitable. So these are the different references for the today topic. Thank you so much. Stay blessed.